Hi, everyone, and welcome to the last webinar of the year for the Healy ALS platform trial. It's really been amazing to have this series. The idea for this series came from our patient advisors, and I just want to thank you for bringing this up. It's really been wonderful for me personally, and I know for many of the other speakers to meet with you on a regular basis. And we really love to answer questions live and also love to provide updates as they happen. So for tonight, I'm gonna keep the updates short because we really want to spend most of the time with a live Q&A with our fantastic guest speaker, Dr. Chibnik from the Harvard School of Public Health. For those of you who have been here before, she already gave us a great presentation on the trial from a biostatistical perspective and answered a lot of questions. Again, as a statistician, clearly doing a much better job than me when you ask statistical questions. So that's why we love having her here, and we will take your questions tonight. So if you have any questions about statistics, please type them in the chat. We received a few beforehand, but certainly we also welcome live questions. And then again, if we don't get to all of them, I'm sure that she will be back in 2021 to answer more questions. So with that, I'll provide a quick update. So if you want to go to the next slide. We are ending 2021 with really, great, excitement about the fact that we already completed enrollment in the first 3 regimens. The 4th regimen is very close to completing enrollment, and it's really been wonderful. We started the trial last year with 3 regimens. In 2021, we added the 4th. And then if you go to the next slide, we can see how we already completed enrollment for the first 3. Number 4 is very close to finish, to the finish line. We have a lot of people in the open label extension. Really, this is an amazing community partnership, and really, we want to thank you as we end the year. This is the holiday period is really a time to give thanks to the people who were really on this mission together. So thanks to all of you, and especially to the trial participants and their families for making this happen. Next slide. And as soon as we're back in 2022, we will be super happy to continue to expand this perpetual adaptive trial. We are going to be launching regimen number 5, or regimen E, in early 2022. And so that's great to have 5 drugs starting in 2022. Next slide. So this is a quick reminder. We will take a break next week and the following, and then we will have, I know our patient navigators, Catherine and Alison, are working on a great schedule for 2022, and we will share it, as soon as we have more details. But again, we will continue to have this series. We know that the second week of 2022, we will have a fantastic guest speaker from the Muscular Dystrophy Association. We are very grateful for their support, and certainly happy to hear from Dr. Sharon Esterly from the MDA. So we look forward to that, and we will have more guest speakers. Keep sending us ideas for webinars, and the links to sign up for the webinar and the sign up QR code for our newsletter are on the slides. And with that, I would like to turn it over to the star of this webinar. Go to the next slide, please. Here she is, Dr. Chibnik, who teaches at the Harvard School of Public Health and is an expert biostatistician. I'll turn it over to you, Lori.
Okay, thank you, Sabrina. It's always such a lovely introduction. I hope I can live up to these expectations. So I'm really happy to be back and answering your questions and talking a little bit about a topic that I love, which is biostatistics. And I realize that makes me a little nerdy. So can you go to the next slide? So I am part of a bigger team of biostatisticians that are involved in this study. Many of us sit here at the Mass General Hospital, and we also work with a group called Berry Consultants that have been really instrumental in building the models that we use to analyze the data in this very unique study. Next slide. One more. So this is a webinar series that was really highly requested, with lots and lots of questions that were sent to us about biostatistics. And just like I love to punt all of the clinical questions to my clinical colleagues, they in turn love to return that favor and send us all the biostat questions. So they asked me to come and help talk with everybody about some of the questions about how to interpret data, what is it that biostatisticians do, etc. I'm going to give my little preamble I gave last week or last time in case people weren't here and they're new. And that is one of the things about biostatistics is our job is to look at groups of people. So I want to see if this group of people is characteristically different than that group of people. That is the entire field, is people who have taken this drug, do they perform different than people who are not taking this drug? It is a privilege to be able to look at groups of people. I realize, though, that as a patient or a caregiver, you don't have that privilege. You're concerned about what we call of n equals 1. So you're less concerned about the group of people and you're more concerned about that person that you care about. And I take this privilege very seriously, and I realize that in everything that I do, those groups of people are made up of individual people that have a very vested interest in themselves or their loved ones. And I try to keep that in mind as I do our statistics, as we work together to make this study successful, and also as I answer questions today. So what I ask of you is that you understand that the way we do statistics and the way we can help the population of people with ALS is to think about different groups. And I, in turn, will remember that all of you watching this webinar are very interested in individual people, in addition to groups. So can we move next slide, please? One more. So this is my version of the slide that Sabrina showed. It is how this study has been run. And I'm only showing this to talk a little bit that says biostatisticians are involved in every aspect of the study, from before, and I do have a pointer that I can use, so let me get that going, or not, maybe not, before they consent to the platform, before the study was even started, biostatisticians were involved. We're involved in building the randomization schedules. We're involved in assigning people to different regimens and building those schedules. We're involved in looking at the analyses, designing the analyses, and moving forward. So next slide, please. So this is just to say that every aspect of this study is thought about by biostatisticians, by clinicians, by everybody, and put a lot of thought into it. So as Sabrina said, we started the study by looking at 3 regimens simultaneously. And within each regimen, we were able to randomize more people into drug than placebo, because at the end of this, once everybody has finished their 24 weeks, we're able to combine these placebos into one larger placebo group and use that as comparison for all the different drug groups. Next slide. I think this is the last one. Is that correct? So I went through this in a little bit more detail last time, and those videos are available. I'm also happy to answer questions. But one of the powers of this study is that we're able to run multiple clinical trials, test multiple drugs by keeping the number of people on placebos as low as possible. That we do that by combining these placebos. So that has been the biggest benefit of this study and this particular study design. So on that note, I have a few questions that I've already see appear. And the first one is one that I get quite a bit, which is, how do you define statistical significance for any of the regimens in the HEALEY trials? And really, what is statistical significance? So very briefly and very simply, statistical significance is the question of, this effect that we see, is it bigger than what we would have expected if just chance was what was going on? So if we just by chance ran the same study and we know that there's nothing going on, is what we found bigger than what we would have found if we had just done this by chance? So if I flip a coin 10 times and I get 9 heads, is that more heads than I would have expected if the coin was a fair coin? And that's really what we're saying when we're saying statistical significance. Now, how do we define this in the HEALEY studies? Are the people that are on the drug performing better than the people that were given placebos? This is measured through ALSFRS-R, and the slope or the trajectory of ALSFRS-R. I do have a problem saying that in one breath. ALSFRS-R. Are the people on the drug not declining as fast as people on the placebo. And we also do take into account survival as well. So for each of our regimens, each of the drugs are compared against the full group of placebos. And if it's bigger than what we would expect to see by chance alone, then it is determined to be statistically significant. This is usually measured by something that I think people are familiar with by now, which is called a p-value. And the p-value is really just the probability that we found what we did by chance alone. So the probability that we found the difference that we did if in reality the drug doesn't work. So a really small p-value, say 0.04, means there's only a 4% probability that we could have found what we did if in fact that drug didn't work. At that point, we feel that the probability is pretty small that it was just by chance that we are going to believe that the drug actually worked. So I hope that answered that, but you can keep submitting more questions as we move along. What does the common statement in a clinical trial powered for blank mean exactly? So that's another excellent question that we get quite a bit. So powered for means, do we have enough people enrolled in this study to be able to show a difference if that difference really does exist? So if in reality this drug works, and we never really know walking into a study, we think it works, that's why we're studying it. But if in reality this drug does work and there is a difference, how many people do we need to enroll in this study to be able to show a difference that's big enough that it's statistically significant? Because remember, even if the drug works, it's not perfect. There's always variability. People are always going to respond differently, and it might work better for some people than for others. So if we only enroll 10 people, that signal or that difference isn't going to overshadow the variability or the noise. So how many people do we need in the study to see that difference and make sure that difference is significant? And a lot of work goes into determining that number. We are very cognizant of, we don't want to run a study that isn't powered to see a difference. We also don't want to run a study with thousands of people if we don't actually need thousands of people to see the difference. So we're very cognizant, and a lot of work goes in to trying to determine this number. And for the Healy study, the number was determined to be 160, or 120 on drug versus 120 on placebo. And again, we share the placebos across the different regimens. Okay, so if I have a score which is ALSFRS, which I use to monitor progression of a disease, why do I have to have a placebo from your point of view, which is, I'm assuming my point of view? That is true. If we have a measure that is used to measure progression, and with ALS, it is expected that people will progress in a negative fashion. So why is it that we have to look at a placebo? Can't we just say, if people aren't progressing in a negative fashion, then the drug must be working? So the question of why do we need a placebo is always one of the more difficult questions to answer, because I realize it's a fairly sensitive subject. I'm going to answer from a statistician point of view, and a trialist point of view, is that there is such a thing as the placebo effect. And the placebo effect has various different things. There are biological placebo effects, but I'm actually going to talk a little bit more about the placebo effect of when somebody is being put on a drug and in a trial, they actually might behave a little bit differently. There is something about when we take a new drug that we think and we move forward and maybe answer questions differently, et cetera. And we make decisions slightly differently. Does that always happen? Not necessarily. But from a statistical point of view, I really need to know that given these two groups are exactly the same, except that this group gets a drug and this group gets a placebo, I need to know what the difference between those two groups are. So I need to know that these two groups have had the identical environment, the identical everything except for one thing, and then I can test whether those two groups are different. And this goes for both the participant getting the placebo, but also the clinician that's working with the person. It's very important that the clinician also doesn't know if that person is on a placebo. Sabrina, would you like to add something?
No.
All right, you're good. Okay.
Just the only thing I wanted to say is that I noticed that there was a raised hand in the audience. So unfortunately, we don't have the with this webinar technology, we cannot bring people in as panelists. So if you have a question and you're raising your hand, if you don't mind writing it in the Q&A box, then that way we can answer. Thank you.
Okay, thanks. Okay. So I'm going to move on to another question, and I do expect that the placebo question will come up again in future seminars, and I will keep talking about it in different ways and why we justify it or why we need them. How do you account for that we all have a different ALS? Do you look for subcategories to find if it worked for some, but if it doesn't work for all? And that is an excellent question. The study, the way it's designed right now, and is looking at everybody who's on drug versus everybody who's on placebo as the primary analysis. That is what we are powered to do, and I'm glad I answered the powered question first, because that's what we powered on. We powered on the primary analysis of drug versus placebo. But the data that's going to come out of this study is going to allow us to look at subcategories as well. We have to be very careful when we look at subcategories, because every time you break up your data into smaller categories or you compare one more thing, you up the chances that you are going to find something that isn't actually real. You're going to find something that might exist in your data set, but really doesn't exist because you're going to find it by chance. So we have to be very careful. So we powered for the primary endpoint, and we're going to use this data moving forward to be able to look for biomarkers, to be able to look at subcategories of ALS to help us design future studies. So using those results to help us design future studies. Also, I could say that there's many people here at MGH Biostats that are also excited to use this data to really look for biomarkers for ALS, which absolutely will help us design future studies as well. So as soon as the last patient has their last visit and this data is available, there is a team of people that are really hoping to learn so much more about ALS from this study. So again, as Sabrina said, thank you all that are participating in the study, because your data is providing information, not just for the drugs in the study, but for future understanding as well. Yeah, and so that actually leads, there's another question I see that's a similar question, so I'll read it out loud. Does the statistical analysis change, or is there any difference if there is a post-study ad hoc analysis by analyzing different groups post-study? Should the p-value for significance change? The answer to your last question is yes. So, and as I said before, which is every time we do a subgroup analysis, especially if it wasn't planned, we're digging deeper into the data to try to find something. It really increases our chance that we're going to make an error, and that error being we say that something is effective when in fact it isn't. So every time you look at another analysis, you're taking the chance that you're making that error. So one way to control for that is we do look for a different threshold for our p-value for significance. There are many studies that power towards multiple endpoints, meaning they might power towards a sub-endpoint, like those post-study analyses. But in that case, you would need a lot more people. So in designing this study, it was more important to be able to run multiple regimens, share those placebos, and then later, as we design future studies, we can use post-study ad hoc analyses to help us think about other hypotheses and other studies as well. Okay, if 120 is needed on drug and 40 on placebo, why does regimen D and future regimen need placebo? Hasn't the placebo already been met? So there's a couple of reasons. There's a couple of reasons why we continue having a placebo group. Some of which is historical placebos aren't always appropriate to use for various reasons, which I can talk about. But another is the importance of blinding. So the clinicians, the research coordinators, the participants, nobody on the study that interacts with participants, including the participant themselves, knows if somebody is taking a drug or a placebo during the randomized controlled trial portion. And that is very important because although many of these measures, we aim for the measures to not be subjective, there's always some level of subjectiveness that can go into either collecting these measures, assessing how people are doing, even if it's unconscious. So what we don't want to do is end up having people say, knowing that everybody's on the drug, so kind of even subconsciously looking for there to be an effect. So it's really important to always have a placebo group for that one reason. Also, another benefit of this study, the more placebos we have, we actually do kind of increase our power a bit, which is nice, because that means we're able to see smaller effect sizes. So if there's a smaller difference between the drug and the placebo, if we have more power, we might be able to see it as well. And whether or not historical placebos can get used is sometimes determined based on the drug itself, based on how treatment of ALS is going to change over the years as well. So that's always something that we're asking, is whether or not historical placebos can be used or whether or not we need to collect new placebos. Okay, so I believe that it's been said that the Healy platform trial is powered to see a 30% improvement in ALS-FRS slope. If that's the case, is it possible to not meet the 30% at P of less than 0.05, but conclude that it has reached an improvement of, say, 20 at a slope of P less than 0.05? Oh, I love that question. Sorry. I love that question because I feel like I'm going to use that question on an exam for one of my stats classes. The answer is yes. Absolutely 100% yes, but let me explain why. So we powered to see a 30% difference with some guess or some educated guess based on previous data at what the variability is going to be in ALS-FRS slope. So we use previous data to make a good assumption of the variability that we're going to see in ALS-FRS slope. And that's how we powered it. 30% with this variability, we want a P of 0.05. If the variability is actually smaller, we are powered to see a smaller reduction. So it doesn't mean that we're not gonna, we might see a 20% reduction. And if that variability is smaller than what we thought it would be, we might even see it at P of less than 0.05. So there's a few different factors that go into power. But the main ones are signal, which in this case is 30%, and then noise, which is the variability. So we want something where the signal outweighs the noise, which is also a book, by the way. It's a pretty good book. Okay. Do you think the size of the placebo effect could be different depending on the route of administration? Was that a consideration in the trial design? Another great question. Wow, I'm always getting great questions at this seminar. Yeah, no, the size of the placebo effect absolutely could be dependent on the route of administration. As you know, of the regimens that we have going now, we have injections, we have pills, we have a liquid or suspension, and RGE or regimen. The next regimen will be an infusion. And we do take that into account. We don't, I kind of presented it in a somewhat simplistic way and say we just mush all the placebos together. We don't, we stratify the placebos in our model, and we do try to take that into account to see if the placebo groups are all behaving similarly. And if we see that a placebo group is behaving, say, better than another placebo group, that would indicate that there's a bigger placebo effect in one route of administration than the other, and we would take that into account. So that's all built into our analyses that will happen as soon as the last patient has their last visit, and we get to start analyzing the data that way? That's a really excellent question. If the Healy regimen were to advance to phase 3, excuse me, if a Healy regimen were to advance to phase 3, would the ratio of placebo to drug be 1-to-1? It doesn't have to be. It's going to depend on how they design the study, how big of an effect we see, how it gets powered. I think everybody is fully aware of the fact that we would prefer to have more people on drug than placebo, especially if it's a drug that we're bringing to phase 3. And it's just going to depend a little bit on how it's designed with the drug company and conversations with the FDA as well. But it isn't necessary that it has to be 1-to-1. If a new regimen with a very large expected effect size gets selected for the Healy trial, would you be able to reduce the size of the active and/or placebo cohorts? That's a question that's a thinker. I think based on everything that I said, the logical answer, and let me finish my full thought, is yes, because if we expect a large effect size, we need less people to see it. But I think we would need to think about this more and discuss it in terms of the Healy platform trial, and being able to consistently do the regimens in a very similar fashion is one of the reasons why we can combine the placebos. So I think it's an answer that I don't have a full answer to, but the question is definitely from someone who was hearing what I was saying on the power question. So I like that. So yeah, if we expect a bigger effect size, or if we expect smaller variability, power-wise, you would need less people, but whether we would do that in the Healy trial, I think is something we'd have to work out a little bit more and think about the pros and cons. I'm noticing that we're at 5:30, and I'm told I'm happy to go a little bit over, but I don't want to overstay my welcome.
Well, you never do. Maybe we can take the last few questions, sort of address them quickly. I know that some of these are very complicated concepts,
Yeah.
so I wonder whether I'm sure we can download all these questions and maybe come back in the new year and have you again address some of these. But maybe, why don't you go quickly over the last few questions to address them as well.
Okay. So I last time somebody was like clicking the ones I answered, and I didn't click them this time. So I'm going to do it as best I can. So the first question, and I feel like I should have answered that first, is a very specific one, that you should only compare each section of the ALSFRS-R and not tally all the sections as a total score. What are your thoughts on this? I also will encourage, based on the end of that question, for you to reach out to one of the coordinators that you're saying that it was scored incorrectly. It measures different things. There's pros and cons to looking at a combined score versus individual subscores for any type of measure. And as I said, I think once the data is collected, people are going to be looking at the different subscores, although the actual study itself is powered on the full score, because that is really the overall measure for ALS at this time, and based on what we have. I think I did that one. Regimens. There is a clinical question that I will give to Sabrina later, who can follow up with. Having over 10,000 in a data set of existing ALS patients, again, is there something significantly different with fresh, air quotes, placebo patients? The answer, unfortunately, is yes. The way ALS has been treated over the last years has changed. So we want to make sure that our placebos are receiving the exact same treatment as our active. It's just not having that drug. And then also, based on a response I gave earlier about route of administration of the drug, that also comes into play as well. That doesn't mean we're not using these existing data sets of 10,000 or more, or previous studies, to help us design the regimens as they move forward and come up with the numbers that we do. Subcategories I did, Healy 101. Sorry, now everybody just started throwing more stuff in here. So I'm going to end on this. Why do cancer clinical trials not have placebos? If true, why can't this be the same for ALS? Cancer clinical trials, you're correct that they don't have a non-drug group. What they have is the standard of care group. So a placebo in a cancer clinical trial is what that person would have gotten if they were not part of this cancer clinical trial. For ALS, that's a similar philosophy as well. It's just for ALS, you know, as you know, many people are on the other medications that they're prescribed by their clinician, but we don't, everybody is on the standard of care. The active group has an active drug, the placebo doesn't have it. So it's just a different way of thinking about a placebo. So I think that pretty much, one last thing, if a drug shows the same effect like the placebo in reducing progression, doesn't that mean that the drug is effective? Because at the end of the day, we can't prescribe a placebo. If you see the same effect in the drug group and the placebo, it doesn't necessarily mean that the drug group is effective. It means that giving people a drug, be it a drug group or a placebo, is having some sort of effect on them. And if they're the same effect, we can't say that the drug has given any benefit over giving somebody a placebo. And it's not ethical for a clinician to just give somebody a placebo if they're not part of a clinical trial. So that's a hard one to answer, except to say that we really don't know the drug is effective. All we know is that something that went into doing that clinical trial, the attention, and by attention, what I mean is the more attentive, you know, you're seeing the clinician more often, possibly for the clinical trial exams. Maybe that is the thing that's really showing the difference. We can't say that without it being different than a placebo, unfortunately. I think there are one or two others, which I'm happy to answer next time.
Or if you want to address them quickly.
The only one that's outside of what we've been sort of talking about, how do analyses account for patients who start in the study group but do not complete the study? We do account for them. All of their data is included in the study, and there are different statistical techniques for handling either dropout or dropout for a reason, if there is an adverse effect, etc. And then finally, why can't you look at and publish interim results along the way throughout the trial? How would that impact statistical significance of the outcome? Every time we look at the results, interim analyses, we increase our chance of making an error and saying it's effective when it isn't. So it would make it harder at the end to show statistical significance if we published interim analyses, because we would base our decisions of moving forward on what we saw in the interim analysis, and that biases us towards possibly saying it's more likely that we will find something significant when in fact it really isn't.
Yeah. So Lori, I think there's a lot of common themes that we may want to develop more fully. I think maybe dedicating a session to placebos, different questions and sub-questions. I mean, there's many different ways of asking similar questions that might be helpful. Maybe one session to placebos and maybe one to the ALS-FRSR and biomarkers. I think, that would be great. So maybe we can work together to have you back.
Yeah, no, absolutely. I'm more than happy to come back. I really have enjoyed, I will say this, the Healy platform trial team is one of the best groups of researchers I've ever worked with. And I'm not just saying that because Sabrina's on the call, but they're very devoted and dedicated and really, really want to find something to help the community. And yeah, it's been such a pleasure to work with this group. So.
Thank you. And likewise, I mean, we have really a large and diverse and experienced, really a creative team of statisticians. Lori, obviously, we love working with you and the other statisticians, and love learning more about these difficult concepts, because you were able to explain some very complicated concepts tonight. So let's definitely do more of these in the new year. I know that there was one clinical question that wasn't addressed, so I think I can take that quickly. And it's about regimen D or pridopidine. So the question was, what's the drug for? And so the drug has pretty robust preclinical data in preclinical models, and the hope is that it will help slow down the disease like the other drugs. And the question specifically asked if there was a preferential effect on bulbar function. I think that's perhaps because of the mechanism that's similar to some drugs like Nuedexta that work on bulbar function. Actually, that's not the only mechanism. So the expected effect of pridopidine would be broader than that, not just on bulbar function, but on the entire function of the body. Obviously, we need to test it, and that's why we have a rigorous trial. We're almost done with enrollment for that regimen. So obviously, as soon as we're done with that, we can shift to the monitoring phase. And as Catherine just posted, thank you, Catherine, there was a science webinar about pridopidine that was posted on our website. So we had some of the representatives from the company and scientists who joined to share more about the mechanism of action of the drug. So refer to that for more explanation. Well, and with that, wow, it's bittersweet. It's great that many people will be enjoying some holiday time with their families, but definitely we will miss this weekly opportunity to interact with all of you, and we'll definitely be here the first week of January 2022. Happy New Year, everyone, and we'll see you soon. Thank you.